Thank you. Welcome everyone. Today I'm going to talk about structural variations between wild and domesticated gene pools of Lima bean. So the Phaseolus genus, popularly known as beans, are a group of legumes native of the Americas, including 80 species distributed across the continent. And among all the Phaseolus species, five has been independently domesticated. We can see those five species here in this table. These five species belong phylogenetically to two separate lineages. So the first group is the vulgaris group, mainly from Mesoamerica, and the second group is the lunatus group, mainly from South America. Now, these species present a high nutritional value due to their amount of proteins and micronutrients in their seeds. And the production and consumption around the world are focused on Phaseolus vulgaris, with an estimate production of 30 million tons per year, followed by Phaseolus lunatus, with an estimate production of 200,000 tons per year. The Phaseolus genus is considered one of the most economically important legume crops worldwide. Now, within these two groups, we can find domestication events and genetic pools. For the wild forms of Phaseolus vulgaris, this has three ecogeographic gene pools: the Mesoamerican M1, the Andean A1, and a third gene pool, a wild Andean, the A2. On the other hand, we can, for the wild forms of Phaseolus lunatus, this is structured into four main gene pools: two Mesoamericans, M1, the Mesoamerican M2, and two Andeans, the A1 and the A2. Now, these two groups can be cultivated in a wide range of zones and climates, and they are adaptable to different ecological changes like heat and drought stresses. And this is a key trait for climate change scenarios. Now, one of the difference between Phaseolus vulgaris and Phaseolus lunatus is that the lunatus, or Lima bean, explores a wider altitudinal range than the common bean, as we can see in this image. Now, genomic research has largely focused on Phaseolus vulgaris, leaving aside or with less emphasis in species like Phaseolus lunatus. So that's why in our research group has contributed to fill this gap and in generating new genetic resources. As we can see here, we constructed a chromosome-level genome assembly for the domesticated Mesoamerican Lima bean, leading us to our current work, the de novo assembly of the domesticated Andean variant genome. So we built a chromosome-level genome assembly of the Andean variety of Lima bean using PacBio Hi-Fi technology, and we generated 17.36 gigabytes of highly accurate circular consensus sequencing with an average genome coverage of 25x. The raw assembly length was 598 megabase pairs, with a high completeness of 98.7 of BUSCO genes detected. Also, the assembly contained 1,070 contigs with an N50 of 8.71 megabase pairs. After the reads reanalyzing with the interspecific genetic map with the previously variant, the Mesoamerican domesticated variant, 132 contigs, adding to 527 megabase pairs, could be assigned to the 11 chromosomes. So in this image, we can see the chromosome-level assembly for the domesticated Andean variant. Here in B, we can see that the placement of the contigs along the 11 chromosomes show that the gene-rich regions were assembled into a single contig. Also, in C, we can see the GC content. The GC content varies between the two regions. And for the gene-rich regions, this is more stable at about 30%. And for the pericentrometic regions, it increased up to 60%. Also, in the H, we can see that genetic distance with the genome assembly of the mesomeric lima bean, showing chromosomal stability. Now, after annotating the transposable elements, we identify 97,581 elements, covering about 48% of the assembly, with the most common be the LTR gypsy, that account about 48%. And we also identify common DNA transposons. And in E, we can see that the regions with the high LTR gypsy concentration were the pericentromeric regions. Now, with this de novo information that we gathered, we wanted to understand better the wild species for the lima bean. So we did a structural variance study. Structural variants are large genomic alterations, and we sequenced wild relatives of the domesticated variants. And we did this with 3 accessions, 2 for the Mesoamerican and 1 for the Andean. So we did the read mapping approach, and we used Oxford Nanopore technologies. So with the long reads datasets, we did a quality assessment using Nanoplot, and then we did the mapping using Minimap2. So we took the raw reads and we mapped them to the domesticated accession. So the Mesoamerican with the Mesoamerican, and also we did the same for the Andean. After that, we get the variance using the software NGSEP for long reads. Now, in this table, we can see some of the results for the 3 accessions. We can see the insertions and the deletions, and the comparisons show no bias between them, but the deletions are more frequent, but they still maintaining a balance. Now, in this slide, we can see some of the results only for the accession, the Mesoamerican wild accession, 250-28. And we can see in these histograms the distribution of the insertion size. And in the first histogram, we can find the sizes between 0 and 1,000 base pairs. And here we can see a peak between 100 and 200 base pairs, showing that the most prevalent structural variants are the shorter insertions. The second histogram shows the distribution between 1,000 and 10,000 base pairs, and they are more variable. And in the third histogram, we can see the large insertions up to 10,000 base pairs, but they are fewer than the shorter ones. Now, with all this information, we wanted to understand a key trait for this species, and this is the cyanogenesis. So we explored the biosynthesis of cyanide compounds, specifically the hydrogen cyanide, HCN. But we focus on Phaseolus lunatus because Phaseolus vulgaris does not produce cyanide compounds. So in this image, we can see a Keck metabolic pathway, and we divide it into 5 main stage reactions. So in the first stage reaction, we can see 4 main pathways to the production of hydrogen cyanide. In this stage, the amino acids transform into oximes with certain type of, or with the help of certain enzymes. And in the second stage, we can see that the oximes converted into the precursors of cyanide. Also, HCN, or hydrogen cyanide, is linked to storage compounds, linomarine. This storage at the end of the third reaction. Now, for example, linomarine in some varieties of lima bean decomposes when enters in contact with water and certain enzymes, and producing hydrogen cyanide as a natural defensive mechanism. So that's what we expect to see at the 4th and the 5th reaction. If you would like to learn more about this subject, or if you have questions about this particular topic, please feel free to visit poster A59, and one of our members will answer all your questions. So we identify some regions linked to the 5th reaction, to the production of hydrogen cyanide. This is an IGB image with one of those regions. This is the 3rd chromosome. We can see here in the first row is the reference genome for the domesticated Mesoamerican variant. And we can see the genes. In blue, we can see the forward, and in red, reverse. These are parallel genes. And in the second row, we can see the transposable elements. And in the two rows, the structural variants with the wild Mesoamerican accessions. These structural variants, as we can see here in this image, are near the gene promoter of the reference genomes. Our hypothesis is that these might influence the gene expression for the production of hydrogen cyanide. Also, this could explain why some wild variants produce more cyanide than the domesticated variants as a natural defense mechanism. But this is an ongoing research. Now, to conclude, these species are crucial for global food security due to their high protein and micronutrients. Phaseolus lunatus is a promising species for adaptation to climate change, and we generate new genomic resources through the assembly of domesticated Andean variant of Phaseolus lunatus, and structural variations likely impact expression levels affecting hydrogen cyanide production. Thank you.
Whether these data are all in the public domain?
I really don't know. No, I'm not sure about that. But I can ask for you.
It would be great to get them there. It would be a fantastic resource for the global community.
Okay, thank you.